schedule page and click right there, you'll have it. The reason I thought I had this up here was I actually had done all this, and I forgot to make the link to this guy. So anyway, that's where the link is there. Okay. I'm a little bit behind, but that's fine. I can handle that. We can handle that. I want to finish up saying just a little bit about cholesterol. I was going through that fairly quickly yesterday afternoon. But I hope what I left you with was that understanding that, first of all, the body has some interesting systems for monitoring how much LDLs it makes. And they're not perfect, and it's one of the reasons that people have high levels of LDL. But when the system is working, it actually works pretty well. And I hope I also convinced you of the need for your body to make LDL, or make things that ultimately become LDLs. All right? That's important because we have to deliver cholesterol to cells. And so if we want to deliver cholesterol to cells, LDLs are the only way we can do that. So though your doctor will talk about LDLs being the so-called bad cholesterol, the fact of the matter is that you need to have them to deliver that into cells. What I didn't tell you yesterday was why your doctor calls the LDLs the bad cholesterol. So I want to spend just a couple minutes talking about that, and then we'll move on to photosynthesis. So LDLs are called the bad cholesterol because it's the levels of LDL that actually correlate with increased incidence of atherosclerosis. All right? The higher your level of LDLs, the more likely you are to have problems with atherosclerosis. So the question arises as to why does that happen? How does that happen? Okay? Well, it turns out that LDLs contain fatty acids that, for whatever reason, are relatively easy to oxidize. They're relatively easy to oxidize. All right? Well, those fatty acids in an LDL, if they get oxidized, can cause some problems. Well, first of all, how they get oxidized. We've talked about reactive oxygen species, and there's the culprit right there. Reactive oxygen species, oxygen molecules that have unpaired electrons. How do we get reactive oxygen species? Well, we get them to a limited extent by normal cellular metabolism. A really good way to get reactive oxygen species is to smoke. Okay? So smoking will increase your level of reactive oxygen species in your bloodstream, and will correspondingly cause your LDLs to have increased levels of oxidation. Those LDLs, when they oxidize, may come in contact with the arteries in which they are traveling, and as a consequence become attached to or cause damage to the artery itself. When that happens, your immune system kicks in and says, oh, there's damage in the body. Got to fix the damage in the body. And so there's a group of immune cells called macrophages that will attack the damaged site. Well, if there's an LDL sitting there, okay, and a macrophage comes along and attacks it, there's likely to accumulate a series of LDLs at that very same site. Okay? You start slowing the bloodstream down because you've got something plugging it. It's more likely like you have a floating log in a stream. Okay, and it gets blocked, it's going to catch some more logs going by. So the same thing happens with the LDLs that are coming by. They get sort of all agglomerated into this area that the macrophages are attacking. And so what happens is you create something that people describe as foam cells. They're foamy because they're loaded with LDLs, and the LDLs themselves are loaded with cholesterol. Of the various types of lipoprotein complexes that I talked about yesterday, LDLs have the highest concentration of cholesterol. So they're foamy. Cholesterol is not very water soluble, so just keeping these guys solubilized, they look foamy, and these foam cells that are arising as a result of this attack can turn into what's called an atherosclerotic plaque. So that's the genesis of an atherosclerotic plaque, that a series of events happen. Ultimately, this may harden and prevent or cause a physical block that's happening in that artery. Okay? So the more LDLs you have, the more likely you're going to have oxidative damage, and the more likely you have oxidative damage, the more likely you're going to form a block. Okay? That's a simple fact of life. Now, the bad news is LDL levels are linked to atherosclerosis. The good news is that you have a class of lipoprotein complexes that are actually linked to good things, as it were. Okay? Reduced levels of atherosclerosis. And these are the so-called HDLs, high-density lipoprotein complexes. Now, these guys are kind of complicated. They are actually implicated in helping to remove cholesterol from the bloodstream. Now, cholesterol and some of these factors you can imagine floating around the bloodstream may get deposited. They may get left behind because of damage to one or another. HDLs scavenge these pieces that are left and take them back to the liver. So one of the things that your doctor will look for when they examine your cholesterol levels, they're going to examine all these different lipoprotein complexes. And if the doctor sees a high level of HDLs, they're probably going to pat you on the back and say, that's really good, because it's the high levels of HDLs that are actually helping to prevent some of these problems that I've just described to you from happening. How do you increase your levels of HDLs? Well, one of the best ways to do it is to exercise. Exercise, exercise, exercise. Okay? Exercise will increase your levels of HDLs. Smoking will decrease your levels of HDLs. So not smoking, obviously, doesn't increase them anymore, but it will prevent you from decreasing them if you do smoke. Okay? So good news, bad news, yin-yang, as it were, associated with those two levels of complex. Okay. Now, this last figure I sort of described yesterday, but I just want to make sure that we're clear on this process that's happening. So I said LDLs were means of delivering cholesterol into cells. You saw this figure earlier in the term. This was a process that I called receptor-mediated endocytosis. Receptor-mediated endocytosis. This process internalizes LDLs that contain cholesterol. Okay? And it happens because there's a receptor on the cell surface that is grabbing a hold of an LDL, bringing it in, and literally taking all the pieces apart so that it can use what's there. This is the mechanism I've been talking about that gets that cholesterol into the cell. Okay. Questions about that? Oh, by the way, I didn't talk about the exam. Everyone know about the exam, right? I almost forgot. So I've decided that the exam will not be comprehensive. Yes. Wow, talk about making a class happy. I should have said that earlier. I got everybody in the right mood to start the day. Okay, that means no note card, all right? But it will only cover material since the last exam. I'm sorry, and it's worth 50%. And it's worth 25%. Material since the last exam. Okay? All right. So that's the plan. Yes?
Will that adjust or affect the curve at all in previous tests?
It will have nothing to do with previous tests. It will just be 25% of your total grade. Okay? All right. Okay. I think we should sing about this. To make a cholesterol. Some things that you can build with acetyl-CoAs are joined together partly thanks to thiolase. They come together, 1, 2, 3, 6 carbons known as HMG, and you're on your way to make a cholesterol. To synthesize a mevalonate in the cell requires reducing HMG-CoA as well. The enzyme is a reductase controlled in allosteric ways when the cell's impelled to make a cholesterol. The mevalonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly to build a pp-geranyl in the cell's routines to make a cholesterol. A single step links farnesyls, but that's not all. The squalene rearranges to lanosterol. From that, there's 19 steps to go before the sterols apropos, which you must recall to make a cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase. And statins mimic, so they say, the look of HMG-CoA. So we sing their praise and not make cholesterol. Okay. All right. On to photosynthesis. Okay. Photosynthesis, you're going to see, has some very close relations to a couple of processes you've already seen something about. The first of these processes is electron transport. And that's because photosynthesis uses electron transport. They use it in a slightly different way than what our mitochondria are doing, but you'll see some very definite relationships. Electron transport, of course, in our cells is linked to oxidative phosphorylation. In chloroplasts, it's linked to photophosphorylation. Okay? So we make a proton gradient to make ATP by using electron transport. All right? Chloroplasts are using a proton gradient to make ATP also. We'll see they do it in a little bit different way, but there's some very definite relationships between the two of them. Now, the second process that is very much like what we've seen so far is the pentose phosphate pathway. We'll see that the dark reactions that I'll describe here have some definite similarity to the pentose phosphate pathway. Well, photosynthesis, of course, in plants occurs in specialized organelles called chloroplasts. I note that photosynthesis occurs in bacteria as well. Bacteria don't have chloroplasts, and there's different considerations for them. I won't go into that here. But I do want to talk about what occurs in plants. Chloroplasts are a little bit like mitochondria in a couple of ways. One, they do have electron transporting proteins and complexes inside of them, like our mitochondria do. They are sites of synthesis of ATP, and they're major sites of synthesis in ATP in plants. And the third thing is that there are only 2 organelles in eukaryotic cells that have their own DNA. They include mitochondria and chloroplasts. These guys have their own DNA. It is thought that the DNA that they carry are remnants of the primordial organism, that is a primordial cell that got engulfed by these various, what cells became plants and later and other ones became animals later during the evolutionary process. Okay? So the fact that they have this DNA is interesting. I won't really say much about it here, but if you're curious, come and talk to me about that. The chloroplast is actually somewhat more complicated than the mitochondrion is. All right? The chloroplast has some regions that I will be describing to you in the overall process of photosynthesis. We see an outer membrane and we see an inner membrane, kind of like we see in mitochondria, but in fact, all of the action of the electron transport happening in the chloroplast are happening in these thylakoid membranes right here. Okay? So it's not happening out here at this region. It's happening deep inside of the chloroplast. That's where the action is really occurring. Okay? The thylakoid space, as we will see, is the sort of equivalent of the mitochondrial matrix. Okay? So the thylakoid space is the equivalent of the mitochondrial matrix, and we will treat the stroma somewhat like we treated the intermembrane space in the mitochondria. Okay. I don't want to show you this except for just to say a couple things about it. First of all, during photosynthesis, what's happening is plants are gathering energy from photons of light that are hitting them, and that energy is used to split water. Water is actually being split. Water is being oxidized. It's losing electrons. It's giving up electrons, it's giving up protons, and liberating molecular oxygen. Okay, so plants, we all know, give up molecular oxygen. The reason there's so much oxygen in our atmosphere is because plants have liberated a good deal of that oxygen during the process of photosynthesis. This process occurs in what we call the light reactions of photosynthesis. There's also a separate series of reactions in photosynthesis called dark reactions, and they're a little bit misnamed in the sense that darkness is not required for them. Lightness is required for the first set of reactions in photosynthesis, but darkness is not required for the second set of reactions. During the second set of reactions, the plants are taking, that is in the dark cycle, plants are taking molecular carbon dioxide from the atmosphere and reducing it. And they're reducing it to make glucose. Okay? So when we think about all this glucose that there is out in the environment, all this glucose that's out there, essentially, it ultimately has its roots in the energy of the process that you're seeing right here. Okay? Chlorophyll is a name for the light-gathering complexes that are found in chloroplasts. Okay? Chlorophyll is the name for that. If you look at this structure, it will look very similar to a structure that you've seen before, and the structure that's similar to this that you've seen before is heme of hemoglobin. Heme is very, very similar to this structure. Okay? One difference, however, is that heme has at its center an iron atom, and chlorophyll has at its center a magnesium atom. There are some other minor differences, but that's the essential component of the light-gathering apparatus. Okay? Now, I don't want to go into that. That will confuse us, so that's not a good idea. Let's talk about what happens at the molecular level now during the process of photosynthesis. Okay? Photosynthesis is described as occurring in a Z-shaped scheme. And if we look at this on its side, it looks like a Z, up, down, up, sort of like that. And these are plotted in terms of energies. Okay? So we actually see some different energies arising from these different actions that are happening. Well, where do the energies come that excite what's happening in this process is that cells are exciting electrons. The energy of the excitement of the electrons is coming from light. It's the photons of light that's actually causing that excitement. Now I'm going to step you through and show you how that occurs in just a second. Okay? But all of the energy here is coming from external sources. Photons of light coming from the sun, coming from a lamp, or whatever. All right? So that's the sources of the energy that's going on here. In the case of our cells, when we're doing oxidative phosphorylation, the electron sources are from reduced electron carriers. Okay? So what we'll see in a lot of ways is that this process I'm getting ready to show you is the backwards of electron transport in us. In us, what are we doing? Let's think about it. We take electrons, we dump them into the electron transport system, and they combine with what at the end? They combine with oxygen and make water, right? So we start with reduced electron carriers. That produces oxidized electron carriers. NADH goes to NAD. And at the end, oxygen is going to water. In photosynthesis, we're going the other way. We're starting with water, okay? And water is actually the source of the electrons, okay? That are being, that's indirectly the source of the electrons that's being excited here. Okay? So water is split instead of being made. The electrons pass through a series of carriers, and as we will see in the end, NADPH is made. So it's kind of the reverse of electron transport, and I'll reiterate that in a bit. So what I want to do now is take some time and go through this molecular process of this capture of energy. That's what we're following here, is the capture of this energy from the sun. It's really remarkable. It occurs in two stages, as we shall see. Something called Photosystem 2, which is the first one, and Photosystem 1, which is the second one. And it's not named that way to be perverse. It just turned out they were studying Photosystem 1 before they studied Photosystem 2 and realized, okay, the order in which we studied them doesn't necessarily mean they're the order in which they work. Okay. What happens? Well, the magic of this process actually starts right here with this guy. This guy is a manganese complex. This manganese complex contains an electron that is ready to get excited. It's ready to get excited. Okay? In this photosystem, this manganese atom is sitting here, and when a photon of light hits it, it gives up its electron. Okay? Well, what does that do? That leaves behind a manganese really, 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 really wanting an electron. It just lost it to a photon. It's gotten excited. That electron is way up here. It's no longer part of the manganese. It's starting a process of traveling that I'll talk about in a second. Meanwhile, here's manganese standing around, and it has no electron. Okay? What does manganese do? Well, manganese is able to take electrons from water. It takes electrons from water. It's the removal of electrons from water that causes protons to be liberated and molecular oxygen to be formed. So we're oxidizing water. It's a remarkable process. One of the things that people are really interested in doing right now are finding catalysts that can mimic this process effectively. Okay? We find catalysts that can do that. We can readily make molecular hydrogen, which can be used for fuel for various vehicles and so forth. Okay? Finding economical ways to do that is a very intense search that's going on right now. Well, plants have it figured out. They've done it with this guy. They excite that. This is left behind. Molecular oxygen is generated. So we see molecular oxygen is generated very early. Bang. There's oxygen. Meanwhile, this electron is traveling on a journey, as I described to you. Okay? This electron is passed from this photosystem 2 to something called plastoquinone, PQ. Plastoquinone passes the electron. Here's this electron carrier. We see the electron moving through a series of carriers, just like we saw in the electron transport system. They have different names, okay? But it's the same basic process that's going on, okay? This is depicted as going downhill because the energy is going downhill, and we know that balls like to roll downhill, so too do electrons like to move from higher to lower energies right here, okay? From plastoquinone, this electron passes into this system here, as we will see, causes some interesting things to happen. That electron passes from there to plastocyanin, which now passes it off to the next photosystem. Okay? So we've gone from photosystem 2 through a couple of carriers. This important carrier here I'll say a word about in a second. Okay? And last, the electron has been deposited now in photosystem 1. In photosystem 1, another photon of light excites it. And when the second photon of light hits this photosystem 2, the electron travels on a new journey. And this new journey eventually carries it to a ferredoxin protein. Ferredoxin is an iron-containing compound. This ferredoxin protein donates electrons to NADP and makes NADPH. So what have we done? Electrons have traveled through a series of complexes, and we've deposited them on the terminal electron acceptor, which is NADP, making NADPH. Now, one thing you didn't see that at least I haven't said anything about so far, is you haven't seen any protons being pumped. That's a difference. And it turns out there is a difference. And it's an interesting one. Okay? In electron transport, complexes 1, 3, and 4 pumped protons. What did they do? They pumped them out of the matrix into the intramembrane space, right? This guy right here, this complex in the middle, it's a cytochrome complex is what it is. This cytochrome complex right here pumps protons also. But instead of pumping them out of the thylakoid space, it pumps them into the thylakoid space. So in the mitochondrion, it would be the equivalent of pumping them from the intermembrane space into the matrix. It's backwards. But the result is the same. We still create a proton gradient. In this case, we have more protons inside of the thylakoid space than outside. Those protons still want to get out of that place where they're in high concentration, and they pass through an ATP synthase that has a little revolving wheel that is almost exactly like the ATP synthase in your mitochondria. Okay? So even though the gradient is opposite, the result is still the same. The products of this process that I have just described to you are that we've made molecular oxygen, we've made, as we will see, ATP, and we've made NADPH. Now, in the case of us, we were capturing energy from electrons that had been used in oxidation, In this case, we're capturing energy from electrons that have been excited by light. In the case of us, we make water, we make NAD, and we make ATP. That's the three products. Here, we're making oxygen using water. Okay? We're making ATP, that's the same, and we're making NADPH as the terminal electron acceptor. What's NADPH useful for? Well, it's very useful for making, what have we talked about that needs NADPH? Oh, God, why does he do this? Huh?
The last thing you talked about.
Cholesterol.
Actually, not really cholesterol, fatty acids. Fatty acids need NADPH. NADPH can also be, through some contorted reactions, converted into NADH. So that can be used to make glucose as well. Okay? So both of these things are useful, as we will see, for the process of making glucose, and that will happen in the next set of reactions called the dark cycle. Before I go to that, I want to ask if you have any questions about this cycle. Yes? This is the whole light cycle. Now I'm going to show you a couple variations on it in a minute, okay? But this is the entirety of the light cycle right here. Okay? Yes, sir?
So I realize the dark cycle isn't necessarily happening in darkness.
Right.
But does the plant, say, stock up on the products of the light cycle and then perform the majority of the dark cycle at night?
It's a good question. So does the plant stock up on the products of the light cycle to do the dark cycle at night? The answer is yes and no. One of the things that we're going to see is that during the light cycle, particularly in the summer, it's so efficient that cells start running out of NADP. If you run out of your terminal electron acceptor, what happens to electron transport in us? A lot of things are going to stop with a high voice. Yes? Right? Yes, sir?
If you don't need dark for the dark cycle, can these two happen concurrently?
Can you what?
Can the light and the dark happen concurrently?
They do happen concurrently. Yes, they do.
You said there was a complex in the middle of the two photosystems?
Cytochrome complex. That's the proton pump. I see some frowns. That must mean there's some questions.
Is there some reason why manganese works great for that?
Is there some reason why manganese works great? Metals in general have fairly mobile electrons. And so there probably is a reason manganese instead of something else, but I don't know. I'm not sure if the answer is even known. But metals have fairly movable electrons. So that's probably the reason why it's a metal. That's why we use metals for conducting electricity. Okay, so those are the steps in the light cycle. Kind of a cool process. Now, this gives us molecular oxygen. The last thing I didn't mention, I'll just show right here. Here's the protons being pumped in through the cytochrome complex. High concentration in. Here's the protons moving out, and there's that ATP synthase. Same thing. That's a different name. We'll call it ATP synthase, and you're fine. Okay? Pretty cool. Okay. That's the same thing, a little bit bigger figure. Same thing you can see there. Here's photosystem 2. Here's our cytochrome complex, bringing in the protons, plastocyanin. This is photosystem 1. There's the ferredoxin protein. There's NADPH being made. There's the protons going back out. Okay. Here's some of the molecules. You don't need to know these, okay? But, well, you should know what they are, but you don't need to know the structures. I just point out the structures to you. Because what do they have? They've got long nonpolar tails. What does that tell you? What does a long nonpolar tail tell you in the cell? What's that? It's hydrophobic. What does that tell you? Where does it exist? In the membrane. It's stuck in the membrane. This guy is stuck down in the membrane. And it's very mobile. It's a small molecule. It moves very readily. We saw coenzyme Q that was very mobile. It moved very readily because it was small. Had a tail very much like this guy has right here. Exactly the same phenomenon going on. Okay. Other quinones play roles. Okay. Now, let's imagine that I am a plant, and it's the longest day of the summer, and it's hot, and the sun is beating down on this plant. Okay? And what's the plant doing? The plant is going through these photosystems. It's kicking all these things through. And in addition, one of the things that the plant is doing is it's running out of NADPH. Why? NADPH is used to synthesize things, and even though the dark cycle, as we will see, uses NADPH is going on, the light cycle is more efficient than the dark cycle is, and basically kicks it off. Okay? So how is it then, if we had the equivalent situation that we have in our cells with the electron transport system, if we run out of an electron acceptor being oxygen, we stop everything, right? Then we're really tied to doing fermentation. Plants are saying, well, here's all this energy, here's all these photons coming down, and if I don't have enough NADP, if I stopped everything, I'm hosed. Is there anything I can salvage out of this process? And the answer is there is. Okay? There is. So when the plants start running out of NADP, they short-circuit the process. They short-circuit the process. Okay? Well, how does this occur? What happens is that there is a leakage that happens as a result of the complex 1 accepting electrons but not passing them off to ferredoxin and NADP to make NADPH. They go through what's called cyclic phosphorylation. Cyclic phosphorylation. Okay? In this case, complex 1, okay, is passing off its electrons backwards through the cytochrome system, and these electrons that are coming backwards are providing the impetus to drive the protons in. This guy isn't donating its electrons off to NADP. It's donating it backwards here and causing protons to come through. The protons coming through allow this system to make ATP even if it can't make NADPH. Now that's really useful. To answer your question, I think it was your question about do these things, or it was your question back here, do these things stock up their things for the dark reaction? In a sense, they're doing that because they're saying, well, I can't make any more NADPH, but let's make as much ATP as we possibly can. So in this sense, they are stocking up, as it were, so they can use ATP and NADPH later, when the light is gone, to make glucose. This makes sense? This is called cyclic phosphorylation. I'm sorry, cyclic photophosphorylation. It's not as efficient, but it still allows the cell to make ATP. All right. This shows it in another way, okay? This guy is now, instead of donating things over here, it's short-circuiting. It's coming back this way and making all the... Actually, this is showing the overall, but this in the short circuit is going through here like this, okay? All right. When we look in bacteria, one of the things that we discover that is similar to plants is bacteria basically do cyclic photophosphorylation. Okay? They basically do cyclic photophosphorylation. They don't have chloroplasts as such, and I won't say more about them, but they do cyclic photophosphorylation, allowing them to make ATP by harvesting energy from the sun. Let's start this guy. Okay, let's talk about the dark reactions. You guys want to memorize this for the final now that you don't have a note card, right? That was a joke. Ha ha. All right. This is what happens in what's called the dark cycle. Yes, sir?
So in that short-circuit reaction, what ultimately happens to the electrons?
What ultimately happens to the electrons is that they are just cyclically going through that complex, from the ferredoxin back through the photosystem 1. It's a cyclic process. Water is not being split anymore. Okay. This is the cycle that's happening that's making glucose. Okay? This cycle is aimed at making glucose. It has many reactions that are very similar to what we saw in the pentose phosphate pathway. And I know I went through those quickly, and I'm going to go through this fairly quickly, but there's a couple things that are unique that you need to understand that are happening in this process that are not happening in the pentose phosphate pathway. This pathway is called the Calvin cycle. It's named for the person who discovered it. Calvin, C-A-L-V-I-N. Okay? Now, as I said, it uses many of the reactions of the pentose phosphate pathway, but there's some unique ones. The very most important unique one is the very first one that happens in the process. Okay? The first one that happens in the process is catalyzed by an enzyme that's unique to photosynthetic organisms, and it's called ribulose 1,5-bisphosphate carboxylase. Big long name. You're probably going to call it Rubisco, R-U-B-I-S-C-O. Ribulose 1,5-bisphosphate carboxylase. Well, the name ribulose 1,5-bisphosphate carboxylase actually tells us what it does. Ribulose 1,5-bisphosphate is the molecule that it starts with. And what does a carboxylase do? It puts on a carboxyl group onto that. Well, ribulose 1,5-bisphosphate has a total of 5 carbons. If I put a carbon onto it, I have 6 carbons. The math is very good. Okay. Now, that makes a 6-carbon molecule that is unstable, and it falls apart into 2 molecules of 3-phosphoglycerate. Where did you see 3-phosphoglycerate before? Glycolysis and gluconeogenesis, right? So if we were to simply take 2 molecules of 3-phosphoglycerate and ATP and NADH and go through gluconeogenesis, we can make glucose, right? The answer is yes. Okay, we could do that. All right? Well, this rest of this pathway is showing us various ways in which the cell ultimately ends up making glucose. That's all that's going on. It's not using gluconeogenesis, but suffice it to say that it could. It could use gluconeogenesis. All right? Well, so what's happening here? All right? 3-phosphoglycerate goes to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate goes to glyceraldehyde 3-phosphate, which goes to dihydroxyacetone phosphate. Now there's some swapping that's happening. These are pentose phosphate reactions, ultimately making glucose 6-phosphate. Glucose 6-phosphate, we can take a phosphate off and we've got glucose, and bang, we're there. How many molecules of CO2 will it take me to make glucose? 6. Okay? It takes 6. So a lot of the numbers you see on here, 12 here, 12 here, 12, 10, 4, etc., these are balancing the equation. Okay? What's the source of the ATPs necessary to do this? Well, the source of the ATPs are, of course, those ATPs that are being made in the light reaction. Notice that in the plant, they are reducing 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate by using NADPH, not NADH. Okay? Where's the NADPH coming? The light cycles of photosynthesis. Okay? Well, the upshot of this is plants are assimilating carbon dioxide, excuse me, assimilating carbon dioxide and making glucose. Now, one of the things that is rather surprising, and I actually get emails by this from people who watch my YouTube videos trying to solve this problem. There's not a solution to it that I know, is that Rubisco turns out to be a very inefficient enzyme. A very inefficient enzyme. Okay? There are problems when Rubisco runs into oxygen, and it makes some things that are less desirable. And this has puzzled biochemists and botanists and a variety of people for many years to understand, how is it that this enzyme is so inefficient, yet it's what virtually all plants use to make this process that makes glucose possible. Okay? How is it this is going on? Rubisco is arguably the most abundant enzyme on the face of the earth. It may be the most abundant enzyme on the face of the earth. And here it is, it's really inefficient. Well, how does that inefficiency manifest itself? Okay? Oh, I had to put this on here.
You said in the presence of oxygen.
In the presence of oxygen, yes. In the presence of oxygen, Rubisco makes a side product. And I thought I had this on here, and I don't have it on here. Okay. Here it is. Here it is. Okay. So when Rubisco encounters oxygen, instead of bringing in CO2, where are we going to have oxygen in the first place? Well, light reactions are generating oxygen. Rubisco, when it grabs an oxygen, instead of grabbing a CO2, makes a phosphoglycerate, a 3-phosphoglycerate, and a phosphoglycerate. And phosphoglycerate is readily converted into glycolate, which gets converted into glyoxylate. Does that ring any bells? The glyoxylate cycle. Where did we see the glyoxylate cycle occurring? Bacteria and plants. And one of the reasons we thought, well, the glyoxylate cycle made sense was it allowed the cell to make glucose from acetyl-CoA. Now we see there's actually another reason plants have glyoxylate cycle. It allows them to scavenge this glyoxylate that's being produced in this byproduct reaction. Kind of cool. Okay? Now, this is happening because Rubisco is doing a non-productive reaction here. It's actually splitting this guy into a 3-carbon piece and a 2-carbon piece. Okay? But plants still make something out of that. They still make something useful because they can use that glyoxylate in the glyoxylate cycle and do things with it. Okay. That's the side reaction. What's the real reaction? Well, the real reaction is this. The real reaction is here's ribulose 1,5-bisphosphate, here's carbon dioxide, here's that unstable 6-carbon intermediate that I told you about. It falls apart in water and makes 2 molecules of 3-phosphoglycerate. Okay. And the last thing I'll say here is, and we're doing photosynthesis pretty quickly, I know, is that the process that I just showed to you happens in all photosynthetic plants. Okay? Now, there are some plants that have readily adapted themselves to fairly dry conditions, and in making this adaptation to fairly dry conditions, they have, in fact, made the process of photosynthesis more efficient. They're called C4 plants. And so I want to say a little bit about C4 plants so you understand what's going on. C4 plants still do everything that I just told you. A lot of confusion about C4 plants. They still have the light cycle. They still have the dark cycle. Okay? The only difference with respect to C4 plants is where they get their carbon dioxide from. Okay? If I have a C3 plant, the reason they're called C4 plants is the first thing that's made in the assimilation of carbon dioxide is a 4-carbon molecule, not a 3-carbon molecule. In the assimilation of carbon dioxide, we saw the Rubisco reaction was giving us 2 molecules of 3-phosphoglycerate. Those are each 3-carbon molecules. That's the C3 part of C3 plants. C4 plants, the very first thing when they grab carbon dioxide is they make a 4-carbon molecule. They make oxaloacetate. But they're only making it as a source of carbon dioxide that they're going to dump into the dark reactions inside of the plant. So I need to explain this to you. All right? A C3 plant sitting there. What does it want? A C3 plant wants to get as much carbon dioxide as it can. It wants to grab it right out of the atmosphere, and it does those reactions right there on the surface of the leaf. And that's the worst place for it to do those reactions. Why? Because the surface of the leaf is where the water is, and water is going to evaporate. Okay? So if the plant can tolerate a lot of water loss, okay, we'll do our dark reactions right here on the surface of the leaf. But what if I don't have a lot of water to let go? Wouldn't it make more sense for me to grab this carbon dioxide for the dark reactions not on the surface of the leaf, but internal to the leaf? And that's what a C4 plant is doing. So a C4 plant is grabbing carbon dioxide out here on the outside, but it's not where the dark reactions are going on. All that this outer layer of leaf cell is doing is grabbing carbon dioxide. There's very little water loss in just this process. It's not doing all this other stuff. Okay? Carbon dioxide gets grabbed. It goes onto phosphoenol pyruvate and makes oxaloacetate. Oxaloacetate goes to malate, which goes into here, which drops off the CO2, and then this cycle just goes right back out here. Now, here's the CO2. Here's the Rubisco. The same thing is going to happen that you've already seen. So all that this is a messenger that's carrying carbon dioxide in. Now, this turns out to save the plant a lot of water, and it concentrates CO2 right here where it's needed. Those two together make C4 plants more efficient at making glucose than C3 plants are. They're more efficient at making C4 plants than C3 plants are. Consequently, C4 plants can do some pretty cool things. Okay, now I'm going through that kind of quickly, so I want to make sure I get through it. Questions about that? Yes? I'm sorry, I can't hear you, Sean. Oxaloacetate is the carrier of CO2. It's converted to malate, but essentially, yes, it's the carrier. That's right. So here we have 3 carbon, 4 carbon, 4 carbon, 3 carbon, 3 carbon, 3 carbon, right? So it's come in here, it's dropped off, it's CO2. Look at what's happening in this process, okay? We have NADPH, it's getting converted to NADP. What are we doing inside of the place where the Calvin cycle is going on? We are making, okay, we are making NADPH, and that's useful for making glucose. Out here, we can have light reactions going on, and what are light reactions making? NADPH. Okay? So it's pretty cool what's happening in this overall process. That's one of the reasons these plants are very, very efficient. They're using the resources that they have, and they're sequestering, or they're actually concentrating carbon dioxide without losing water. Very, very cool process. And let's see. There's all the reactions. How about a song to finish the day? To the tune of Scarborough Fair. Photosynthesis is divine, fixing carbon using sunshine. It's thanks to plants that we've got a prayer. They pull CO2 from the air, reaping energy from the sun. It's efficient, second to none. You grab the photons almost at will. Protoporphyrin chlorophyll. Light reactions of system 2. Split up water making O2. Electrons pass through schemes labeled Z, pumping protons gradiently. ATPs made due to a shift of the proton spinning quite swift. an enzyme turbine cellular maze, you know as ATP synthase. Carbons fixed onto a substrate, ribulose 1,5-bisphosphate. Rubisco acts inefficiently, splitting it into 3 PGs. If the enzyme grabs NO2, it makes glycolate, it is true. The Calvin cycle works in a wheel, giving plants a sugary meal. So photosynthesis is divine, 'cause it happens all of the time, from dawn to dusk and times in between. Solar panels truly are green. Okay. Tester in my office. Just follow me back, yeah. Let's see, I'll be just a minute. So either everybody was tired or nobody knew that song. I don't know if it must have been. Yeah. It's an old, actually it's an old war song is what it is.